Gel electrophoresis is a method that uses an electric current to separate DNA fragments based on charge and size. A typical apparatus setup consists of a gel tank with electrodes that are connected to a power source. The tank is filled with a buffer solution, and embedded in the solution is the agarose gel used for DNA separation. Let's start by looking at how a gel is prepared. First thing is you'll need to determine the percentage of gel you're going to use. This is based on the DNA fragments you're planning on examining. Here is a table you can use for your reference. A 1% gel is usually sufficient for many applications. Once that's determined, you're ready to start making your gel. A gel is made by measuring the appropriate amount of agarose to dissolve in the amount of buffer solution you're planning on using, and that is based on how big of a gel you're planning on making. Once your solution is prepared, melt the agarose by placing your solution in the microwave, but be careful, it can get really hot. To ensure a good quality gel, make sure all your agarose is melted. Check that there are no residual agarose crystals left. Next, allow the solution to cool down, just enough so that it's safe to the touch. Then add DNA stain. This will let you visualize your DNA later on. An example of a DNA stain commonly used is CyberSafe. Make sure to mix your solution well and pour it into a gel casting tray to solidify. Don't forget to include a gel comb, which is used to make the wells to load your DNA samples. Once solid, the gel is placed in the tank that is then filled with the buffer solution. The orientation of the gel is key. Make sure that the gel wells are on the side of the negative electrode. This is because DNA is negatively charged, and when a current is applied, it will move towards the positive electrode. Basically, you don't want it running off your gel. So here is a tip to remember. DNA runs to red. Now, let's understand how DNA is separated based on size by taking a microscopic look at the gel. Once solid, the agarose forms a 3D gel matrix with channels and pores of different sizes. That way, small DNA fragments will travel through the gel faster than larger fragments. So now that we have a good understanding, let's put all this together. Say you ran a PCR and you want to check 1. Was it successful? And 2. Did you copy the region you're interested in? So is it the correct size? Remember, before loading your sample, you want to mix it with a loading dye. This lets you track the DNA as it moves through the gel and increases its density so that it stays at the bottom of the well. And in addition to loading your DNA sample, you will also load a DNA ladder solution, which is made up of DNA fragments of known sizes and is used as a reference to determine the size of your DNA on the gel. Once everything is loaded, let your samples run through the gel, and now you're ready to analyze them under UV light. Let's say in the ladder, this DNA fragment is around 3,000 base pairs long. Then your PCR product would be around that size. If that's what you expected from your PCR, then hooray, your PCR was successful.